Hi, today we'll show you how electroeluting is performed. In order to purify DNA fragments, such as digestion and PCR products for further purposes, like cloning and sequencing, electroeluting can be one of the methods of choice. Based on the negative electric charge of DNA, a high sound barrier is placed within the electrophoretic field, trapping the fragments and preventing their further migration. This method allows the purification of a wide range of DNA fragment sizes from different sources, with a relatively high efficiency of up to 80%. Its low costs represent another advantage of electroeluting when compared to other purification methods, such as columns, resins, kits, among others. The DNA sample is first visualized by a titanium bromide stained agarose gel electrophoresis in order to visualize and identify the desired fragment to be purified. Electrophoresis should proceed until fragments are well separated and their size can be precisely determined. Following electrophoresis, the desired fragment is neatly excised from the gel using a brand new razor blade and taking care to trim the gel slice as close as possible to the band of interest. This procedure should be performed with a proper protection from UV light and wearing gloves. After filling the electrolyter with the appropriate burning buffer and taking care not to spill it over the surface of the middle plateau, the gel slice is placed inside one of the wells, as close as possible to the V channel.